So I just want to give a little, very, very much of an overview about genomics from the way that I see it. And I think there are sort of some quite significant drivers at the moment, which is leading to implementation of genomics into mainstream practice. Clearly, it's the genomic technology that's a major driver, and particularly the cost of genomic technology. So at the time of the Human Genome Project, it cost $100 million to sequence the genome. And now they're talking about sequencing the genome for probably less than $1,000. So it's really, really quite powerful. The second is the change to the infrastructure that supports the technology. And clearly in England, that's happening through NHS England with their reorganisation of services. So they have what are called genomic medicine centres, which is where the laboratories and clinicians are going to work together to have a look at this sequence data. And then there's the consumer. So there's going to be greater opportunity now to basically buy over-the-counter genetic tests. And the relevance of that to primary care is going to be interesting in terms of how we manage that data that comes into practice. So I believe that debate question is absolutely right. I think we should be early adopters because I think we should be able to help to influence all the parameters that are going to help us deal with our patients who want information about genomic knowledge. So this is about sequencing. So, and I think the first thing to say about it is the first three overall boxes are basically going to be black boxes to us. We don't particularly need to have great knowledge about the choice of sequencers or the sequencing chemistry or even how the data analysis is done. Suffice to say that it's complicated. But then what we've also heard is about point of care testing, that genomic technology is going to be increasingly available. And I think there's no reason why we can't have a benchtop sequencer in practice. Maybe federational practices might consider this as cost effective, but I think it's going to happen. And there's an example already of an instrument produced by a company called Oxford Minion, and they basically can connect up their sequencer to a USB port on the back of your computer, and you can do sequencing in practice. So I think it's going to be really interesting. The other really important aspect of this slide, though, is at the end, because once you've done all the data analysis, you need to classify the gene variant that comes through. And this is going to rely on multidisciplinary teams making a judgment based on the evidence. And there's plenty of evidence out there in terms of genomic databases, in terms of whether a particular gene variant that's been identified is going to be pathological or not. And even at the level of the multidisciplinary research teams or multidisciplinary teams, I think primary care have got a role. And in South London, for example, where I'm a GP, we're going to have a GP, hopefully on an MDR team. And I think it'd be quite interesting to see what influence a GP has around this. So I said that the driver was genomic technology. The second is about the infrastructure. So the 100,000 Genome Project is really sort of driving the impetus forward in terms of genomic medicine at the moment in England. And the three areas that are being particularly looked at are around cancer, rare diseases, and infectious diseases. And there's been, with regard to cancer, so lots of work going on around germline mutations, but also somatic mutations, so mutations within the tumour, and how that you can look at biochemical pathways, you can look at new targets for treatment, and these are coming through. So, for example, in melanoma, for example, there's a really good drug that's been shown to be quite spectacular in terms of response to patients with melanoma. Rare diseases has been quite successful to date as well. So we all know about the diagnostic odyssey that families have to go through before a diagnosis of a rare disease is made. And the diagnostic yield through the 100,000 Genome Project has been around 27% to 30%. So it's looking really encouraging in terms of being able to help these families come up with a diagnosis that they can then subsequently manage. And then infectious diseases, and this is because, particularly for example with viruses, and in this case HIV, because of the studies that you can do around RNA, gives you lots of powerful information around studies around HIV and resistance in particular. And then the other driver I mentioned was about the consumer. So you can now walk into a particular retailer and buy a kit for, I think it's about £100, and you send off a mouth swab, and about 6 weeks later you get a printout back which gives you information about complex conditions, such as asthma or hypertension or diabetes. It can give you information about personal traits, and it can also give you information about ancestry as well. But then there's also a sealed envelope in one particular company who offer this, in that in this sealed envelope, if you go into it, it will tell you your risk of dementia, of Parkinson's disease, and of whether you're a carrier for BRCA, for the breast cancer disease. And this is all without pre-test counselling. So the consumer can go to the commercial company and get this. When the results come through, what does the consumer do? Well, they could come back into general practice, or they could seek advice from secondary care. And Nadeem and Judith and I and others wrote a paper a while back about what perhaps needs to be thought of when these tests are done, and when a patient may present to you with this information. So you want to know basically how robust the data is, what level of risk increase there is before the test is deemed useful. Do the variants interact with other variants, and what difference does that make to your risk? What are the particular interventions if you come up with a gene variant for a particular condition? What's the evidence? What information is given to the consumer before and then after testing as well? So clearly at the moment, these are really challenging questions, based on the information that comes through from this testing. And I think I'm probably right to say, Julian, the secondary care might even struggle with some of the information that comes through here. So
the first thing is the understanding of what is a variant and what is a mutation is little understood. The second thing is that we're getting more information about things like these SNPs, which came through the breast cancer work, and they're not even necessarily in genes. So the understanding of how the SNPs and the environment impact on our genetic risk is a whole new area which is being looked at, as well as pharmacogenetics. So it's so many different layers of complexity that it's that education and understanding which is going to be difficult.
Great, thank you. So lots of challenges ahead of us in terms of trying to keep abreast of all this, and Judith will tell us later about some of the educational interventions that might happen in general practice to help support this. Clearly, there are going to be new types of clinical pathways that we're going to have to we will interact with, and so, for example, the knowledge around inherited cardiac diseases and new clinical pathways are available. GP informatics, we've heard about that this morning, about linking genomic information with longitudinal health data is going to be really important. We've heard about cost effectiveness today as well, and as Julian also has mentioned, about accuracy and reliability of the information coming through. So undoubtedly there's a lot of challenges ahead in terms of adopting this into practice. So as this is sort of predominantly a research meeting, I just wanted to cover 3 or 4 different areas that I think are going to be sort of relatively important for primary care, just as exemplars of how genomic medicine may help us. So we heard about the pharmacogenetics around warfarin. One of our major challenges in practice is around polypharmacy and multimorbidity. And although we have lots of regulatory information about drugs, pharmacoepidemiology, it's how all these drugs interact is something that we're really quite interested in. and whether genomics may offer new biomarkers that help us sort of manage our patients with multimorbidity. There are actually there's a whole consortium set up that's gathering the evidence around each individual drug that has a genetic, that may have genetic variation in terms of metabolism, elimination, and so on. So pharmacogenotyping is something that might well happen in practice. There are exemplars. So, for example, there's a GP in British Columbia, Martin Dawes, who's developed a system of actually testing a panel of gene variants against particular drugs. And Munir was telling me this morning about the work that he's doing in Liverpool. Again, a panel of variants that help in looking at drug metabolism. So here we have a potential solution in terms of trying to reduce adverse effects, trying to improve outcomes based on evidence-based guidelines with perhaps clinical utility of the genomic information coming through. So the main sort of issues that we need to think about around that are the utility, the efficacy, what predicts for toxicity, and how you can link it to outcomes. And Munir gave good examples of that with warfarin. Again, around pharmacogenomics, this is a sort of like thematic framework that came out of a small research project around for an MSc dissertation. And this was asking GPs what they thought were the challenges of implementing pharmacogenomics into practice. So there were 8 main themes that came through, and some various sub-themes that came under each of those themes. So the utility of the information, the importance of mainstreaming genomics, working with secondary care, the importance of education, the value of the genomic information, so for example, efficacy, helping predict for toxicity, patient factors, so the importance of shared decision making, personalized medicine, which is what we all talk about, ethical, legal, and social implications, the economics of implementing this, so the cost effectiveness, and informatics was really important. So how we record this information, how we code it, how we share it, how we seek consent, for secondary uses of the information as well. So there were some really useful messages that came out of this, really. So the second area that I think we could help with in primary care is around prescribing and infection and antibiotic resistance. These 2 discs are supposed to represent 2 different cells, bacterial cells, and it's amazing actually what microbials do with their DNA. They can swap it around and add to it and subtract to it, which then gives rise to resistant clones, and those resistant clones happen because it affects various aspects of the way that the drug interacts with the bacteria. So I think genomics is really important here in terms of identifying resistance and also gaining new knowledge about how we might tackle antibiotic resistance. But also in terms of infection control, there's lots of work going on in terms of, and again, genomics can be really helpful here. So Public Health England do mandatory surveillance for particularly MRSA and E. coli, and also Clostridium difficile. And they increasingly are going to be using whole genome sequencing to look at sort of the bacterial information that's coming through. We have at the RCGP influenza surveillance through the RCGP Research Surveillance Centre, and this is where GP practices, and there are probably about 200 practices now associated with the centre, make weekly returns to the centre around influenza. So this is an early warning system in terms of trying to identify influenza in the community. And here you could easily see that adding bacterial, mouth swabs and doing genomic analysis could work really well in terms of supplementing the information that's coming through. And then the other really important area is about outbreak management. And this is sort of looking at how transmission occurs. And there's this concept called phylogenetics, which is really interesting. It's about looking at the geographical spread of resistant clones in the community and in different populations. And I'm not just talking about UK, I'm talking about worldwide. And there's been fantastic work that's been done around cholera, for example, where they've defined all these resistant clones around all these different countries. But with shared populations, so for example, between patients who are in care homes and NHS trusts, there's the opportunity to look at these resistant clones and how transmission occurs. And there was a really interesting piece of research that was done in Cambridge where they looked at a neonatal baby unit and they looked at staphylococcus infections as well. So I think primary care offers a population study database here in terms of infection control. And then the other really important aspect of work that we do in primary care is obviously manage complex conditions. And how can genomics help us, with these patients who have these conditions? Well, obesity is one good example, perhaps, and in this case, this slide is showing the year-on-year increase in the prevalence of severe obesity. And severe obesity is defined as a BMI of greater than 40 kilograms per meter squared. So a real challenge. Now, clearly not all of this is down to genetics, but where genetics does help is that it helps us, or genomics might help us, is help to define new gene variants that may contribute to clinical conditions. So, for example, Ensembler is a database which has characterized a number of genes. Decipher is a really important database which is predominantly set up to look at developmental delays, but associated with these conditions have been, is obesity and really severe obesity. So there's lots of information coming through about gene variants for this complex condition, and you can correlate that, you can match that for lots of other common conditions as well. So once you know what sort of phenotype you have, you can then look back and say, well, is there a genotype that actually explains this, or even the other way around? And then you start to get different types of pictures involved. So obesity is not just obesity, but you're going to have subclasses of disease. And that's going to be an extra complicating factor to all the other conditions that we manage in practice. So we're going to have different classes of hypertension as we do different classes of asthma and so on. So it just starts to get even more complicated as we talk. And then the last thing I just wanted to mention was about stratifying the population for risk. This is a study that we're doing with the Institute for Cancer Research, which is looking at genetic profiling to guide prostate cancer screening. And the aim here is to look at the association between a genetic risk score, which is based on looking at over 170 SNPs, single nucleotide polymorphisms, and matching that against parameters such as PSA, biological samples, but also pelvic MRI. And what you do is, before I come to that. So this is a study that's based in general practice. So there are 7 practices involved. And this is where the local CRNs have been really helpful in terms of helping to support the practices, recruit patients, or identify and recruit patients. So it's been really good. And we're looking for 300 patients, and we're almost there now. But this is how the stratification works. So of the 300 men aged between 55 and 69, you do the SNP profiling, and then the top 10% of those, based on genetic risk, then go forward for looking at other investigations such as PSA, MRI, and so on. And then they might go on to prostate biopsy, and then you're looking at outcomes. So what's the final outcome from this? So I think the nice thing about this study is that it's based in general practice, and it may well have potential utility in the future if the genetic score correlates with the outcomes that you expect. So our aim really is how we can mainstream genomics into practice with a GP who is literate in genomic medicine to help meet what the potential demands might be on our system. And I think if I had a little wish list of how genomics might help us in practice, it's about sort of having clear patient pathways where there's been cost-effective commissioning, where we know that actually this is actually going to be really useful for the patient. It'd be really nice to have early diagnostic tools, which includes protein and other types of biomarkers that help us in terms of early diagnosis. Stratifying care, stratifying genetic risk, I think will happen more and more. And also dealing with some of the real challenges that genomics offers in terms of ethics, legal and social, which I haven't really covered at all today. But for example, the down screening test now, which is down to the use of fetal DNA in terms of trying to identify high-risk pregnancies, is a really good example of some of the ethics and some of the social problems or issues that we might face. So it would be nice in the future that when we look at a genomic database, and this is one such genomic database, that actually it's a database that's going to be easily accessible to general practice, that provides a repository of gene variants that are relevant to a general practice population. And I think that's where the clinical utility and the usefulness of genomics might happen. So finally, I just want to thank the Barcode study collaborators, and this is the Barcode team. Thank you very much. Thank you.